For 4 billion years, life on Earth has been running on a single operating system. The same molecular code, 4 chemical letters, arranged in sequences of mind-bending complexity, has governed every creature that has ever drawn breath. From the first single-celled organism that flickered into existence in ancient oceans, to the 8 billion humans alive on this planet right now. That code is DNA. And for most of our history, we were simply passengers, reading the code, trying to understand it. But something extraordinary has happened in the last decade. We didn't just decode the book of life, we learned to edit it. In 2012, a molecular tool called CRISPR-Cas9 arrived, and it changed everything. For the first time, scientists could cut DNA at any location they chose, with startling precision. It was, as Nobel laureate Jennifer Doudna put it, like having a find and replace function for the genome. But that was just the beginning. Today, we have CRISPR 2.0, tools so precise they can change a single DNA letter, one chemical out of 3 billion, without cutting the strand at all. We're growing miniature human brains in labs. We're engineering organisms with synthetic genomes that have never existed in nature. And we're seriously discussing how to adapt human DNA for life in deep space. This is the story of what happens when we stop reading the blueprint and start rewriting it. What does it mean? What's at stake? And who gets to hold the pen?
To understand where we are, we need to understand what CRISPR actually is. Imagine your genome as a book with 6 billion letters. CRISPR-Cas9, the original version, is like a molecular search and scissors system. You give it a specific sequence of letters, and it hunts through the entire genome until it finds a match. Then it cuts. And when the cell repairs that cut, scientists can insert new genetic information, or simply disable a gene entirely. It's powerful, but it's not perfect. The original CRISPR can make off-target cuts, snipping the wrong sections of DNA. And the act of cutting itself can sometimes trigger cellular alarms, causing unintended consequences. Enter CRISPR 2.0. Base editing, developed by David Liu at the Broad Institute, doesn't cut DNA at all. Instead, it chemically converts one DNA letter into another, like using correction fluid on a single character in that 6 billion letter book. It's being used in clinical trials right now to treat sickle cell disease and certain cancers. Then there's prime editing, sometimes called a molecular word processor. It can not only swap letters, but insert or delete entire sequences with extraordinary precision. In 2023, researchers used prime editing to correct a disease-causing mutation in human stem cells with over 90% efficiency. And the field is moving fast. In 2024, the FDA approved Casgevy, the first CRISPR-based therapy for humans for sickle cell disease and beta thalassemia. Patients who had suffered debilitating pain crises their entire lives were, after a single treatment, functionally cured. Think about that. A single treatment, one molecular edit, and a disease erased, not managed, not treated, erased from a person's biology. That's CRISPR 2.0. But even that is not the most extraordinary place this technology is taking us. What if editing genes isn't enough? What if you want to write entirely new ones, or build a genome from scratch? That's synthetic biology. In 2010, Craig Venter's team announced they had created the world's first synthetic cell, an organism with a completely artificial genome, assembled from scratch in a lab and inserted into a living cell. They even encoded a watermark into the DNA, a quote from James Joyce, the scientists' email addresses, and the names of the team members. Life authoring itself. Since then, synthetic biology has exploded. The Human Genome Project 2, also called GP-Write, aims to synthesize a complete human genome from scratch by the early 2030s. Scientists are engineering bacteria to produce insulin, to consume plastic waste, to detect cancer biomarkers in the gut, Companies are programming yeast cells to produce spider silk, vanilla flavoring, and even experimental cancer treatments. But one of the most startling developments is something entirely different: organoids. An organoid is a miniature, simplified version of a human organ grown from stem cells in a lab. Kidney organoids, gut organoids, and yes, brain organoids. By 2023, researchers had grown brain organoids with functional neural activity, tiny blobs of human brain tissue, smaller than a pea, generating the kind of coordinated electrical signals we associate with thought. Scientists have even connected them to robotic systems, where organoid-based neural networks controlled basic movement. No one seriously claims these are conscious, but the question is no longer completely absurd. Organoids are revolutionizing drug testing, disease modeling, and our understanding of conditions like autism and Alzheimer's without ever touching a live patient. They are, in a profound sense, personalized biology, grown from your own cells, reflecting your own genetic makeup. The line between studying life and creating it has officially blurred.
Here's a problem no one ever had to solve before. If humanity is going to become a multiplanetary species, if we're going to live on Mars or travel to other star systems, we face a biological crisis that has nothing to do with rocket fuel or life support. The human body was designed for Earth, gravity, atmosphere, magnetic shielding. Remove those conditions, and our biology starts to fail. Bones weaken, eyeballs deform from cerebrospinal pressure, radiation from cosmic rays tears through DNA, dramatically increasing cancer risk. A 6-month journey to Mars would expose an astronaut to radiation levels equivalent to 15 to 20 chest X-rays every single day. So, a growing group of scientists, including researchers at NASA and academic institutions, are asking a radical question. What if we don't adapt our spacecraft to protect humans? What if we adapt the humans? The concept is called space genomics, or directed evolution. Using gene editing, researchers are exploring how to enhance DNA repair mechanisms, boost radiation resistance, reduce bone density loss, and even incorporate genetic variants from organisms like the tardigrade, the near-indestructible microscopic animal that can survive in open space. In 2025, a research consortium announced the first successful transfer of a tardigrade-derived radiation protection gene into human cell cultures, showing a 40% reduction in radiation-induced DNA damage. We are not simply exploring space. We are considering engineering explorers who are biologically built for it. The question is no longer, can we do this? The question is, should we? In 2018, a Chinese scientist named He Jiankui announced that he had created the world's first gene-edited babies, twin girls whose embryos he had edited using CRISPR to make them resistant to HIV. He called them Lulu and Nana. The scientific community was appalled. He had made germline edits, changes that would be inherited by every future generation of those children's descendants. He had done it in secret, without adequate ethical review, on children who could not consent. He was sentenced to 3 years in prison, but the line had been crossed. Here's the core tension. Every technology powerful enough to heal is powerful enough to harm. CRISPR could cure genetic disease, or it could be used to create biological weapons, or to build a market for designer babies that only the wealthy can access. Brain organoids raise questions about consciousness, suffering, and the rights of lab-grown tissue. Space genomics raises questions about whether future engineered humans would even be the same species as us. Experts are not of one mind. The National Academies of Sciences recommends strict international oversight of germline editing. The WHO has established a global registry to track human genome editing research. Most countries prohibit germline editing in clinical settings, but enforcement is uneven, and the technology is spreading fast. What's certain
is that the ethical frameworks we have were built for a world where evolution was not a choice. That world no longer exists. We need new rules, new conversations, and perhaps most importantly, broader participation in who gets to make these decisions. Science moves at the speed of discovery. Wisdom needs to keep pace.
We began this story in the cosmos, in the idea that every atom in your body was forged in a dying star, that the universe spent 13.8 billion years building the conditions for life, and then life spent another 4 billion years building you. That blueprint, your genome, is the most complex, most beautiful, most tested piece of software in the known universe. And now, we are editing it. Not randomly, not through the blind chance of mutation, deliberately, purposefully, with intent. CRISPR 2.0 is giving us a scalpel where we once had a sledgehammer. Synthetic biology is letting us write new chapters in the book of life. Organoids are letting us read those chapters in miniature. Space genomics is asking us whether the next chapter of human evolution might take place not on Earth, but among the stars. The question isn't whether we will rewrite the code of life. We already have. The question is who we will become when we do. The universe wrote the first draft. We're holding the pen now. Use it wisely.